All right, so we are going to talk about computer-aided drug design as a part of computers and statistics. That is module number 2, applications of computers in pharmacy. Okay, so let me start with what is meant by medicinal chemistry. Now, why do we need to talk about medicinal chemistry? Because computer-aided drug design is used to maximum extent under the auspices of medicinal chemistry. Okay, and medicinal chemistry and pharmaceutical chemistry, these are two different terms. All right. So, for instance, someone might just ask what is medicinal chemistry and what is pharmaceutical chemistry? What's the difference? So, medicinal chemistry basically is that subject which deals with drug design, drug synthesis, drug evaluation. But pharmaceutical chemistry would be a broader term. Okay. So, in medicinal chemistry, we focus on drugs, how to go about drug design, how to go about finding newer drugs, discovery of newer drugs. But pharmaceutical chemistry deals with discovery of drugs with respect to, the entire gamut. Like, for example, we'll think about, how the solubility properties or the physicochemical properties of the drug should be in order for the for a pharmacist who is specialized in pharmaceutics, that is formulation chemistry, how that person is going to uh, you know design some formulation for this particular drug on the basis of whatever drug we are discovering. So when we are discovering a drug, when you are trying to design a drug, we'll try and incorporate all those properties, physicochemical properties, the toxicity, and all those factors will take into account, and then we'll try to design a drug which is going to be good from formulation point of view. Okay, not just formulation point of view, but from the animal studies and human clinical trials point of view as well. So in a previous lecture also we said, a person who specializes in pharmacology, all right. So that person is going to look into two aspects of drug discovery. One is preclinical testing and nothing but animal studies. And the other aspect is human studies, and clinical trials. So clinical trials and preclinical testing, and human trials as well as animal studies. So we need to, when we are trying to design a drug, when we are trying to discover a drug, we have to think of all the possibilities so that we can discover a drug, we can design a drug that it goes on to be, successful not only in preclinical trials, that is animal studies, but also in human trials, that is clinical trials. Okay. So that is what is the difference between pharmaceutical chemistry and medicinal chemistry. So in medicinal chemistry, we'll deal with drug design, drug discovery, but from the point of view of finding some active molecules. Whereas in pharmaceutical chemistry, it is nothing but the greater side of drug discovery and drug development. So we'll think about drug discovery from the point of view that those pharmacists or those other people who are involved in the drug development for that particular drug will also find it easy to bring it from that NCE stage right up to the drug stage, which is ready to be marketed. Okay, all those animal and human trials are done, and all those analysis studies are done, and then further, a good formulation is made out of it, which is bioavailable. So that overall aspect would be pharmaceutical chemistry. Okay, so that's the difference between medicinal chemistry and pharmaceutical chemistry. So if we have to talk about both of these are nothing but the discipline at the intersection, the discipline at the intersection of chemistry, especially synthetic organic chemistry. Why synthetic organic chemistry? Because we talk about drug synthesis as well, right? And pharmacology. So it is chemistry plus pharmacology and various other biological specialties where they are involved in design, synthesis, and development of pharmaceutical agents or bioactive molecules. That is nothing but the drugs. So basically, in one sentence, medicinal chemistry is nothing but drug design, synthesis, and preliminary evaluation. Now, if someone is still thinking as to why we have started with medicinal chemistry, so यहाँ पे हम लोग ये बात करें ना ड्रग डिजाइन की. So drug design is an integral part of medicinal chemistry. Okay, and drug design can be done in many ways. For example, combinatorial chemistry. For example, molecular modeling. Okay, and there are many other ways. The classical approach of maybe synthesizing a drug, finding out its activity, and then devising a hypothesis for it. So those are all ways of drug design, but उसमें से one of the parts is computer aided drug design. Okay, so that is nothing but CAD. That is nothing but CAD. Okay, so that's the reason why we have started with the broader perspective, and now we'll go on, more and more sharper, more and more focused on what would be computer aided drug design. So when we start with this approach, what happens is our focus becomes more and more clear as we go ahead. Okay, all right. So let's move ahead. Then, what is drug discovery? So, drug discovery would be defined as the process by which new candidate medications are discovered. Okay, so it is the process by which new candidate medications are discovered. So, if you have to talk about drug discovery, see, when we say in the previous slide, what we said was medicinal chemistry is drug design, drug synthesis, and drug evaluation, right? The preliminary evaluation. But if we have to talk precisely, then using the term drug is the final outcome of it, right? What we are trying to do is we are trying to discover a drug, but actually what we are doing is we are trying to discover an NCE. Okay, so NCE is nothing but novel chemical entity, right? NCE is nothing but novel chemical entity. So once the novel chemical entity is produced, is discovered or designed by a medicinal chemist, then it goes on to those clinical trials or preclinical trials, and then it is all those other experts come into picture. Like for example, the analysis people, okay, the pharmaceutical analysis people or the pharmaceutics people. Okay, and then it is sent for regulatory approval. So that means the FDAs are involved in there then. Okay, so for instance, we know that the strictest FDA, the FDA which is involved in approval of most of the drugs is the US FDA, right? And in India also we call it as FDA. So it is Indian FDA. For US it is US FDA. So the Food and Drug Administration, that is going to take care of the approval process. So they will take care of each and every minute aspect of it. They'll look into the purity of the NCE. They'll look into the impurities that are possibly present in the NCE. So, for example, if the pharmaceutical company which is applying for NDA, that is new drug application, if they file that their NCE that they used in their formulation was 99.99% pure, then what will FDA do? FDA will ask whether the drug is for chronic use or if it is for short-term use. If it is for short-term use, then it is still okay. But if it is for chronic use, definitely they are going to talk about the 0.01% impurities that are present in the NCE. Okay. So for instance, if we have everyone, I'll just request you to mute your audios. Yeah. So for instance, I was just talking about FDA is going to ask about the impurities that are present. So 0.01% impurities, if they are present, then what are those impurities? Whether they are going to be carcinogenic impurities or whether they are going to be some impurities that can induce some other side effects or toxic effects, or whether they are, normal impurities which may not affect the health of the human beings who are going to consume that drug. So that is what will be checked, and the benefit of that drug as compared to, for example, placebo. Okay, so clinical trials are designed in such a way that the FDA gets all of the data that is required for the drug and for the formulation. So looking into all of those aspects, FDA is then going to approve the drug, and only after the approval will it be called as a drug. ठीक है, तो ये पूरा का पूरा cycle है. Okay, so we have NCE, that is novel chemical entity or new chemical entity. Then all those trials, preclinical, clinical, all those in vitro and in vivo tests that are done, studies that are done. Then followed by the analysis of the drug, the analysis of the formulation that is made by the Ceutics people. Okay, and then it goes to FDA. The new drug application is filed, and only after looking into all of the parameters that are related to the NCE, FDA will call this NCE as a drug. So ड्रग डिस्कवरी जब हम लोग बोल रहे हैं, ड्रग डिजाइन जब हम लोग बोल रहे हैं, वी आर टॉकिंग ऑफ दिस अल्टीमेट ऑब्जेक्टिव, okay, of discovering a drug. but actually it is NCEs that are discovered, and only a few of those NCEs get converted or are rather approved to become drugs. Okay. So here also, when we are talking of the term drug discovery, we are talking of the entire term, okay, the end objective of our process. So drug discovery is the process by which new candidate medications are discovered. Okay. So let's move ahead. So let us have a look at the historical perspective of drug discovery. So historically, drugs were discovered by identifying, let me just change the pen color. Yeah, so I guess this color must be now visible to all of you. Yeah. So historically, drugs were discovered by identifying the active ingredient from traditional remedies. Okay. So, for ages, we have so many different, alternative systems of medicines that are there. For example, in India, we have Ayurveda. In Western countries, in some of the Middle Eastern countries, there is Unani. Okay, and so on and so forth. There are so many other alternate systems of medicines that are available, and the historically what was done was drugs were discovered by identifying the active ingredient that is present in that were present in those traditional remedies. Or the other way of discovering drugs was serendipitous drug discovery. Serendipitous yani by chance. For example, penicillin. We all know how Alexander Fleming discovered penicillin, right? So that was a serendipitous discovery, yani ki by chance discovery. He was trying to do something else, and he found out that penicillin happens to be an antibiotic. It stops the growth of bacteria, right? So that's how historically drugs were discovered. So substances, either as crude extracts or purified chemicals, were screened for biological activity without knowledge of the biological target. So in other words, हम लोग क्या कह रहे हैं कि traditional remedies में से in the form of crude extracts or the purified chemicals, they were screened against various biological targets for biological activity. Okay, so whatever was their biological activity, it was found out by screening them against a large number of biological targets. Okay, only after the active substance was identified was an effort made to identify the target. यानी कि target identification was the last step. सबसे पहला was, the first thing was to identify whether the given substance has some kind of activity, and then understanding via which target, via which receptor or enzyme that drug is acting. That was the last thing that was done, that is identification of the target. Okay, and then this approach is known as classical pharmacology, okay, or forward pharmacology, or phenotypic drug discovery. All right, so this approach wherein the drugs were found out, or the purified chemicals or crude extracts were taken, they were screened against a large number of targets, and then उनका activity पहले check किया जाता था, and then they used to go and try and find out the targets that were involved. That is the end. That is the receptors or the enzymes that are involved in the activity of the drug. So this approach was known as the, or rather, it is still known as the classical pharmacology approach, or the forward pharmacology approach, or the phenotypic drug discovery approach. Okay. Now, the idea that the effect of the drug on the human body is mediated by specific interactions of the drug molecule with biological macromolecules such as proteins or nucleic acids or enzymes in most cases, led the researchers to conclude that individual chemicals are required for biological activity of the drug. So now the researchers, they started thinking that instead of using crude extracts, we should be using individual chemicals, because it is individual chemicals that are going to go inside the body. They are going to be absorbed, they are going to be bioavailable inside the body, and then they are going to interact with the proteins or the nucleic acids or the enzymes, that is the biological targets with which they want to interact, right? So the central theme now started shifting to finding out individual chemicals, okay, to finding out individual chemicals rather than finding out a mixture of chemicals. So this led to the beginning of the modern era of drug discovery, wherein pure chemicals instead of extracts, okay, instead of crude extracts of medicinal plants, became the standard drugs. So आजकल के जमाने में हम लोग crude extracts की बात नहीं करते. In some systems of medicine, the crude extracts are still used. Okay, for example, in Ayurveda, extracts are used, bhasmas are used. There are various different those different types of formulations that are present in Ayurveda, which are used even today. But as far as modern system of medicine is concerned, we talk about pure drugs. Like for instance, in allopathy, we talk about, say for instance, paracetamol as an antipyretic drug, जैसे बुखार कम होता है, right? So, if we have to talk of paracetamol, we'll talk of the purity of that individual chemical that is called as paracetamol. We are not going to talk about some extract from plant which is going to help us for antipyretic activity. यानी कोई प्लांट का एक्सट्रैक्ट ले लो, उससे बुखार चला जाएगा, right? That is not something that we are going to think of. We are going to think of that particular constituent of a plant extract that is going to be pure and that is going to become a standard drug. So that is the modern era of drug discovery. So examples of drug compounds isolated from crude extracts. For example, अभी पहले के जमाने में crude extracts यूज किया करते थे, like crude preparations यूज किया करते थे. Nowadays they are extracting the active chemical. For example, we have morphine. Okay, so morphine we know is the active agent which is present in opium. Then we have an example of digoxin, which is a heart stimulant which originates from the various species of digitalis, especially digitalis lanata, right? So now in modern medicine, we talk about specific chemicals, okay, specific individual chemicals instead of the extracts. All right. So organic chemistry, the developments in organic chemistry also led to the synthesis of many of the natural products which are isolated from biological sources. Like for example, morphine. Okay. Initially, it was thought कि morphine इतना complex, इतना बड़ा molecule है कि उसका synthesis करना is difficult. Okay. But later on, they went on, the advances in organic chemistry that have happened, the researchers, the organic chemists, they went on to synthesize such big natural products in lab. Okay, instead of isolating them from biological sources, many times they are isolated in very minuscule amounts, right? But agar organic chemistry say we try to synthesize them, then we find, we can have larger yields of those products. So that will make the entire process very effective. All right. So that is how modern era of drug discovery has been enhanced or stimulated or motivated by the advances in organic chemistry. Okay. So later on, small molecules were synthesized specifically to a specific target, a known physiological or pathological pathway, avoiding the mass screening of banks of stored compounds. So पहले क्या करते थे? ड्रग डिस्कवर करने की कोशिश करते थे फ्रॉम क्रूड एक्सट्रैक्ट्स, राइट, फ्रॉम क्रूड एक्सट्रैक्ट्स. एंड फिर एक बार ड्रग मिल गया, फिर उसके बाद में उसका रिसेप्टर ढूंढते थे, right? So from crude extracts, drug used to be discovered, and from then after the efficacy was shown, only after that the receptors were found out. But now what they do is they try to find out which receptors are involved in a specific disease, either physiologically or pathologically. Yani in the normal processes or in the abnormal processes, right? Or in the abnormal processes. So in the normal processes, if a receptor is involved, that is called as physiological pathway, right? And if it is involved in the abnormal processes, then it will be called as a pathological pathway. So now in modern drug discovery, what do? We try to find out the receptor first, the target first, and then for that receptor, we try to devise a drug. Okay, then for that receptor we try to devise a drug. ठीक है. So पहले क्या करते थे, ड्रग से रिसेप्टर, तो दिस वाज कॉल्ड एज फॉरवर्ड फार्माकोलॉजी. लेकिन अभी क्या करते हैं, पहले रिसेप्टर टारगेट करते हैं, उसके ऊपर ड्रग्स डिजाइन करने की कोशिश करते हैं. दैट इज कॉल्ड एज रिवर्स फार्माकोलॉजी. Okay, that is called as reverse pharmacology. And such approach has led to great success. Example, work of Sir Elion and Hitchings on purine metabolism. The work of James Black on beta blockers and cimetidine. Cimetidine, we know, is a H2 blocker. A classical example of how SAR studies have been done for discovery of newer drugs, right? And then discovery of statins by Akira Indo. So these are all examples of modern era of drug discovery wherein receptors were thought of first, the targets were thought of first, and then the drugs were designed for it via reverse pharmacology, right? So another important approach started with developing chemical analogs known as the active substances. So, for example, Sir David Jack et al. From Glaxo, which was previously called as Allen and Hanbury's, they pioneered the first inhaled selective beta 2 adrenergic agonist for asthma. They also pioneered first inhaled steroid for asthma. Then they also went on to discover ranitidine as a successor to cimetidine, right? And then they also worked on the development of triptans, right? So many different wonderful classes of drugs were discovered just because of the modern way of drug discovery, that is reverse pharmacology. यानी कि पहले receptor के बारे में सोचो, first think about the target and then think about designing drugs for that target. Okay? Then, Sir Jedru de Leon, when he was working mostly with a group of fewer than 50 people. See, try to understand this. Now, here we have talked of Sir Elion, right? We are rightfully calling him Sir. Okay. So, Sir Elion, he was working on purine metabolism. He went on to discover so many drugs just working with a group of 50 people. And just imagine those drugs are still used till date. तो वो जो 50-51 लोगों ने जो उस टाइम पे काम किया वो आज तक यूज हो रहा है. ओके, वो आज तक यूज हो रहा है. एंड व्हाट वी सी इज millions or rather billions of people, human beings all over the world have been helped. Their diseases and disorders have been cured just because of the work of this 50-51 people. Imagine that. Okay, so that is the power of reverse pharmacology. So working with mostly a group of fewer than 50 people on purine analogs, he contributed to many discoveries like the first antiviral agent, the first immunosuppressant, the like for example azathioprin. So that was the first immunosuppressant. Okay, so that allowed human organ transplantation. So we know immunosuppressants, they are not good drugs, but they are absolutely necessary when we have to talk of organ transplantation. Okay, so for example we hear of organ rejection. You must have all heard of or maybe read of kidney transplantations that fail. क्यों फेल होता है? बिकॉज किडनी ट्रांसप्लांटेशन ठीक से हो गया, बट आफ्टर दैट द बॉडी ऑफ द रिसेप्टर, डोनर ने डोनेट कर दिया रिसेप्टर के बॉडी में, वो किडनी को ट्रांसप्लांट कर दिया, बट व्हाट हैपेंस नेक्स्ट? द बॉडी ऑफ द रिसेप्टर स्टार्ट्स प्रोड्यूसिंग इम्यूनिटी अगेंस्ट द ऑर्गन, यानी कि immune response against the organ which has been transplanted, because that will be considered as a external thing, right? Xenobiotic, as we call it as. So it will be considered as a external thing to the body, and our body will take it as an alien, and it will start producing immunity, antibodies against that organ. So that organ will start, it will be, our body will start destroying that organ which has been transplanted. So that leads to organ rejection. So azathioprine, it was discovered as an immunosuppressant, and then human organ transplantation surgeries went on to become, larger number of surgeries started becoming a success. So imagine, first immunosuppressant was discovered by again these 50 people. The first drug to induce remission of childhood leukemia, yani ki it was previously called as, initially people used to say cancer is not a curable disease. But these 50 people, they came together, they worked on those purine drugs, and they discovered a drug which could induce, the first drug which could induce remission from leukemia. So लोगों को फिर, they started becoming confident. Yes, cancer can also be cured. Okay. Then they also worked on some pivotal anti-cancer treatments, anti-malarials, antibacterials, agents for treatment of gout. So just imagine, so many different drugs were discovered just by the group of those 60 odd people. And what did they do? They did modern drug discovery. And how did they do modern drug discovery? By reverse pharmacology approaches. Okay, by reverse pharmacology approaches. As simple as that. All right. So that is how drug discovery has progressed. Okay, so this process is called as reverse pharmacology. This process is called as reverse pharmacology. Okay. And later on, we know that human genome has been sequenced. Okay. So in the 2000s, they started sequencing the human genome under the human genome project, and that project is now complete. We know of all the genes of all the proteins that are produced in the human body, right? So that human genome project has allowed rapid cloning of human proteins and hence the synthesis of large quantities of purified protein targets. So this has further enhanced reverse pharmacology. Okay. And now it has become common practice to use high throughput screening of large compound libraries against isolated biological targets, which are hypothesized to be specific or disease modifying. So just imagine the way we have progressed. पहले ड्रग डिस्कवर करते थे, फिर उसका रिसेप्टर डिस्कवर करते थे. That was called forward pharmacology. अभी क्या करते हैं? पहले रिसेप्टर डिस्कवर करते हैं, फिर ड्रग डिस्कवर करते हैं. Okay, and that is called as reverse pharmacology. That is called as reverse pharmacology. Okay, so drug to receptor से अभी receptor to drug back side में हम लोग जा रहे हैं. So that is called as reverse pharmacology. All right. So that is how we have went on to with the history of drug discovery. Now hits from these screens are further tested in cells and then in animals for efficacy, and needless to say, then they are tested in human beings. All right. Now recently, for COVID-19, we all kept on hearing drugs for emergency approval, right? The concept of drugs for emergency approval. So drugs for emergency approval are those drugs which are tested on human beings first with very limited or absolutely no animal studies. So see, it is imperative that animal studies are done first. Okay, so that is what is done for normally approved drugs. But for emergency use, they have clauses that even if the animal study, the amount of animal study, the data from animal studies is restricted, then human studies can be done. The clinical trials can be done on human beings, and then the drug can be approved for emergency use for global pandemics as was COVID-19 that was caused by SARS-CoV-2, right? So that's how we have progressed over the years as far as drug discovery is concerned. Okay, so modern drug discovery, it involves identification of hits from screening, and it involves medicinal chemistry, and it involves optimization of those hits to increase the affinity, selectivity, that is to reduce the potential side effects, efficacy, that is nothing but potency, metabolic stability to increase the half life of the drug, and oral bioavailability. Okay, so sometime back I was talking about the difference between medicinal chemistry and pharmaceutical chemistry, right? So medicinal chemistry is nothing but drug design, drug discovery, and drug evaluation. And then what is pharmaceutical chemistry? Pharmaceutical chemistry is nothing but medicinal chemistry plus optimization of the hits to increase affinity, to increase selectivity, to increase efficacy and potency, to increase metabolic studies. I mean stability, and to increase oral bioavailability. So when we take into factor all of those other things in addition to medicinal chemistry, it is called as pharmaceutical chemistry. So once a compound that fulfills all of these requirements has been identified, the process of drug development can continue. So the other set of people come in, the other set of experts, the pharmacology experts, the ceutics experts, the analysis experts, all of them come into the picture then. Okay. And if successful, the clinical trials are developed for the identified compound, and then it will be approved as a drug. Okay. So I have to now talk about the economics of drug discovery, but I think I'll start with it in the next section of our lecture. Okay. So as of now, I'll just stop here for the time being. And so here we have talked about what is medicinal chemistry, what is pharmaceutical chemistry, what is drug discovery, and how drug discovery was done in the earlier period. And now nowadays in modern drug discovery era, how do we do go about it? Okay, so we'll talk about the economics of drug discovery in the next section. Okay,